Good afternoon, everyone. Welcome. I just want to welcome everyone to our open session. We have folks in the audience who are listening in on the webcast. Welcome to you as well. My name is Charmaine Royal, and I am faculty at Duke University, and I'm one of the co-chairs of this committee. And I'd like to introduce my co-chair, Aravinda Chakravarty. Aravinda?
I want to add my thanks to Charmaine. I don't want to take up too much of your time, except to introduce myself, that I'm a human geneticist who's now a professor at the NYU School of Medicine, and we've long been interested in this subject. And not only to the panel from the NIH, but also to all of you listening on the webcast. Thank you very much. If you have additional questions, you can obviously write to the staff and through us, even after this webcast. Thank you.
All right. Thank you, Aravinda. And thanks to the members of the committee who have agreed to serve. It's a big task we have ahead. And so we're really grateful to those who've agreed to serve on this. Also want to thank the NIH for sponsoring this study and for taking the time to speak with us today. We're going to hear from the sponsors at the NIH about the task at hand, what they're expecting of this committee to do. But before we hear from the NIH, I'm going to read the National Academies disclaimer for open sessions. As co-chair of the Committee on the Use of Race, Ethnicity, and Ancestry as population descriptors in genomics research, I would like to welcome everyone to the open session of the committee's first meeting. I want to note that this is an open, on the record, information gathering session. That is, the committee is in the process of assembling materials that it will examine and discuss in the course of making its findings, conclusions, and recommendations. Therefore, I ask everyone here today to be extremely mindful of the fact that the committee has made no conclusions and that it would be a mistake for anyone to leave here today thinking otherwise. Comments made by individuals, including members of the committee, should not be interpreted as positions of the committee or of the academies. For those of you not familiar with the committee process, this is the first of 5 planned committee meetings. Some of these meetings will include public sessions, where we'll have the opportunity to hear from many experts in the field and receive public testimony. The committee will deliberate thoroughly before writing its draft report. Moreover, once the draft report is written, it must go through a rigorous review by experts who are anonymous to the committee, and the committee then must respond to this review with appropriate revisions that adequately satisfy the Academy's Report Review Committee and the chair of the National Research Council, the NRC, before it is considered an NRC report. The expected report release is early 2023. Further information on this study, including future meetings and the full statement of task, can be found on the project website. The committee is asked to do 2 primary things. 1, to review and assess the existing methodologies, benefits, and challenges in the use of race and ethnicity and other population descriptors in genomics research. And 2nd, to develop feasible and logical approaches to advance appropriate use of population descriptors in published genomics research studies. The open session today provides an opportunity for the sponsor of the study to address the committee and describe the context and reasoning that went into the statements of task so that we can better understand the scope and the purpose of this study. In the interest of time, we will not introduce all of the committee members, but the full roster and their biographies are available on the study website. Today, we will first hear a presentation from Dr. Eric Green, Director of the National Human Genome Research Institute of the NIH, who will present the committee's official charge. After Dr. Green's presentation, we will also hear from a panel of speakers from the NIH, including Mr. Vince Bonham, Dr. Stephen Shannock, Dr. Eliseo Perez-Estable, and Dr. Sherry Schooley. There are 14 NIH institutes, centers, and offices supporting this study, and a complete list can be found on the study website. After the speakers have provided remarks, we will then have time for discussion with the committee. It is my pleasure to invite Dr. Green now to present the charge to the committee. Eric?
Well, thank you so much, Charmaine, and thanks to Aravinda as the other co-chair for your willingness to help lead this remarkable committee through this important set of deliberations. So I'm Eric Green. I'm director of the National Human Genome Research Institute at the National Institutes of Health. On behalf of the 14 NIH institutes, centers, offices, and programs that have come together to co-sponsor this consensus study, I want to thank the co-chairs, the committee members, and the National Academy staff for your time, your commitment, and your dedication in taking on this consensus study. Now, as NIH is the sponsor of the study, I'm going to walk through the charge for the committee, and then I'm going to go and ask my other NIH colleagues who have joined me to answer a few questions before we turn this back to the co-chairs for a broader discussion. Those NIH colleagues include Mr. Vince Bonham from my institute, the National Human Genome Research Institute, Dr. Sherry Schooley from the All of Us Research Program, Eliseo Perez-Estable from the National Institute of Minority Health and Health Disparities, and Dr. Stephen Chanik from the National Cancer Institute. Now, before we move into what the committee will be tasked to do, I think it would be good to revisit why this study is needed. As the committee members are all familiar with, the use of race, ethnicity, and ancestry population descriptors in biomedical and genomics research has been a topic of debate within the scientific community. Recent advances in genomics, which has included new knowledge regarding differences in the prevalence of genomic variants across human populations, have heightened the challenges in the use of social constructs to describe groups and populations. Meanwhile, the rapid growth in the United States and internationally with respect to research programs that involve large cohorts of human participants and extensive genomic analyses has brought further urgency to grapple with how we as scientists describe human population groups. Now, the NIH has been convening meetings about these issues since 1997 and has funded numerous consortia and research studies to understand human genomic variation, such as the kinds of studies and meetings that are illustrated on this slide here. And for many years, we've grappled with the relationships between human genomic variation and the social constructs of race and ethnicity. 2004, NHGRI held the first meeting on race, ethnicity, and genomics in medicine and biomedical research. And then over the last 20 years, NIH has funded a number of scientists and scholars to study research questions related to social identities and the use of race, ethnicity, and ancestry in human genomic variation and genomics research. Now, in 2016, I can tell you that the National Human Genome Research Institute and the National Institute on Minority Health and Health Disparities held a workshop on the use of race and ethnicity in genomics and biomedical research. And this workshop included discussions about the use of Office of Management Budget or OMB categories in biomedical research, also additional data types needed to enhance understanding of race and ethnicity, guidelines surrounding how to collect and report race and ethnicity data, and the need for a global paradigm for describing populations. While the workshop report actually recommended that a National Academy consensus study be conducted, and that is why we find ourselves here today. Now, since then, we have gotten, because of the importance of the topic, 14 NIH institute centers, offices, and programs to come together to co-sponsor this consensus study. I've been at NIH over 28 years. It is not easy to find an issue that you can get 14 institutes to all come together on, contribute funds, and in a unified view, recognize the importance of the topic that is worth gaining additional insights about. But we did it with these 14. And listed here are the 14, as Charmaine pointed out. You could also see it on the website. And while representatives, you're going to hear today on the panel from about 4 of these components, there are representatives from all the other NIH components who are listening into this discussion virtually. Well, collectively, we at NIH are excited that today has arrived and that we are kicking off this important consensus study. So what is the charge for the assembled committee? Well, the committee has been convened as a group of experts from the fields of genomics, medicine, and social science to review and assess the existing methodologies, benefits, and challenges in the use of race and ethnicity and other population descriptors in genomics research. There are 11 tasks associated with this study, which I will just quickly read through so you have them, but this is all available as well. 1, document and evaluate the variety of population descriptors currently used in genomics research and the potential benefits and challenges of changing these descriptors. 2, assess how race and ethnicity and genetic ancestry are currently being used as population descriptors in health disparities research to study genetics and genomics. 3, assess the appropriate use of race, ethnicity, and genetic ancestry as population descriptors in the determination of genetic risk scores and health risk overall. 4, develop feasible and logical approaches to advance appropriate use of race and ethnicity and alternative population descriptors in published genomics research studies. 5, examine the potential of new culturally responsive methods and common data elements, in other words, CDEs, for enhancing harmonization of population descriptors in large genomic studies in the United States and globally. 6, assess when it is appropriate to use race and ethnicity as population descriptors in genetic and genomics research, and provide recommendations to scientists and researchers for future research. 7, propose best practices for domestic and international harmonization of population group descriptors. 8, assess the scientific knowledge of the relationships among race, ethnicity, and population genetic variation. 9, identify and discuss potential obstacles to implementation of the new methods to describe populations. 10, discuss potential implementation strategies to help enhance the adoption of best practices by the research community. And finally, identify obstacles and propose best practices in the use of population descriptors with legacy biological samples and associated data. Now, what are we expecting from the consensus study? Well, in short, we want a final report. And that final report should describe best practices on the use of race, ethnicity, and genetic ancestry and other population descriptors in genetics and genomics research as formulated by the committee. Attention should be given to how these best practices could be used by biomedical and scientific communities to increase the robustness of study designs and methods for genetics and genomics research in the United States and globally. So in closing, I should emphasize that this National Academies study has the promise to guide research practices for the scientific community, to strengthen the entire research ecosystem, and to increase the robustness of genomics studies in the United States and globally. NIH stands ready to help the committee in any way that we can so that we can collectively realize that promise. With that, I'd like to turn now to my NIH colleagues to respond to a few questions that we received prior to today's session. And so with that, I'm going to stop sharing my screen, and I'm going to ask Dr. Sherry Shuley of the All of Us Research Program the first question. Now, I discussed what the tasks are and what the final report should include, but what are your thoughts about what is in scope and what is out of scope for this study and why?
Thanks, Eric. Yeah, I think you did a really good job of explaining the 11 tasks that are within scope of this, this report, but there are 4 things that we actually called out specifically that are beyond the scope of this consensus study. The first is examining the use of race and ethnicity in clinical care. So we're specifically talking about research. The second is examining racism in science and genomics. The third is examining the use of race and ethnicity in biomedical research more generally, so beyond genetic and genomic research. And the last, providing policy recommendations to NIH and other government agencies.
Thank you, Sherry. Vince Bonham from the National Human Genome Research Institute. How do you expect the report will make an impact? You've been involved from the beginning in formulating the momentum for this report. What are your thoughts about its long-term impact?
So thanks, Eric. So this is the first study of this magnitude to bring together a group of experts from diverse disciplinary backgrounds. And one of the things I'm just so excited about this committee is the perspectives, the experiences, the years of thinking about these questions, but coming from different backgrounds. So the potential impact here of having different parts of the scientific community coming together to help guide this is extremely important. The second comment I would make about the impact is this is an opportunity to help to share to the broad scientific community, not just NIH grantees, not just individuals in a specific consortium, but individuals across the scientific community that are conducting genetics and genomics research. And the final comment I would say about impact here is the legacy of the National Academies and the work that's been done over the years and some of the seminal reports that have been published by the National Academies. I think that there's an opportunity here to have such a report that can help guide the future of science in this area, guide policy, guide NIH, guide others. But this broad impact on the scientific community is what I hope for impact.
Thanks, Vince. Eliseo Prestable from the National Institute of Minority Health and Health Disparities. I'd be curious to get your thoughts about who is the expected audience of the committee's report or audiences?
Thank you, Eric, and thanks for the the outlining of all the tasks. In many ways, I think although it is a big project, we have defined, or the group has defined, or the advisory committee has defined, sort of a narrow path. And I think that's it's a challenge because no one's going to look at this as a narrow path. And so I think the committee needs to reflect on that. So the audience is going to be all scientists who work with anything related to genetics, obviously, and also anything related to social epidemiology, minority health, health disparities, health equity research. And I think in many ways, the audiences are different and they're looking for different things. What will be a position on what could be considered either a past controversy or a current controversy? What's the role of genetics in ascertaining influences on outcomes, on clinical outcomes? How does that interact with individual characteristics, whether they be demographics as defined by self-identified race, ethnicity, socioeconomic status, and other, as well as contextual in terms of where one lives, the environment, the policies around you. It's really hard to separate all these things into separate lens completely. And so I think that the challenge is that everyone's going to be paying attention to it, whether they're genetics researchers looking for guidance on how I should do this, A, B, or C, make sure that I follow what the guidance is, to policymakers and consumers who are going to say, well, what's NIH or what's the National Academy saying about genetics and race ethnicity? And so there's no avoiding, I think, controversy in this report. I think we can stay within certain lanes, but I think everything has to be put in that context that it's going to have a much broader audience than one would like to think, that we can tailor it to a small group. So,
thanks, Eliseo. And lastly, I'd like to hear from Stephen Chanik of the National Cancer Institute. Stephen, what communities will likely use the report?
I'd like to, first of all, thank the Academy for agreeing to do this and for this marvelous group of experts to address, I think, what will be a set of critical observations and recommendations that have pleiotropic implications across our whole society. And I think Eliseo started to touch upon that in saying that there are going to be different groups that are going to want to look at the output of this and use this, whether they're researchers or whether they're advocate advocacy groups or whether they are commercial entities. So I think, the charge is starting with a very important academic base to sort of getting the foundational understanding of what and how to address these questions. But I think we can envision that this is going to be very important for the academic community, not only going forward, but the challenge of how to capture legacy information and what may need to be reinterpreted or redone in a way so that it's harmonized, so that we can have the most effective, robust analyses. So the methodologists and the geneticists and the epidemiologists, the molecular epidemiologists and the clinical trialists are all going to be looking for ways in which we can use the past and at the same time go forward with really robust, wise studies going forward. And I think the commercial world is going to see the same way. I mean, the question of how you get to the FDA for a drug, and are there, with precision medicine and precision prevention steps, beginning to really take shape, how and in what way will this information be really critical in some of those determinations? And that's going to get to some very, very important and sometimes very difficult decisions that we're going to have to see. I think the general population is going to be interested in this. We have, recreational or commercial genetics in the sense of people doing this for many different reasons beyond just going to and working with a health provider per se. I'll name a few, but I won't name them all. 23andMe, Ancestry.com and the like. They're going to be looking very closely at this again and potentially reinterpreting what they have against or with what the recommendations of this group are. I think the news media and the legal worlds are each going to eventually want to look at this. I think questions of how studies are reported and what their value is clearly a part of our fabric in both the communications and sort of 24/7 news world. And that will potentially, I think, also have legal and political ramifications. So I think the answer to the question is I think the entire community is going to see this, but with through some different Polaroid lenses. And I think that's going to be the key issue in trying to be able to keep the academics on track and have others understand exactly really what the meaning and the utility of the recommendations are. I think the more practical and the more structured the recommendations, the more constructive they will and helpful they will be in the long run.
Thanks, Stephen. So at this point, you've heard from each of the NIH panelists. I want to turn the Zoom room back over to Charmaine and Aravinda as the co-chairs. And those of us from NIH are now available to answer any questions from the committee or that come in from the public side of this meeting. So we're here and are happy to help.
All right. Thank you so much, Eric and colleagues from the NIH. Again, thanks for sponsoring this study and for really laying out what it is that you're expecting. We have some questions, and you laid out those 11 tasks, Eric, and many of our questions look at some of those tasks, but also look at what you listed as being out of scope. And so the committee members have some questions, and we're going to go ahead and start. Anne, you wanted to ask your question.
Sure. Thank you so much. And thanks to all of you for joining us. I'm Anne Morning. I'm a sociologist and demographer at NYU. And I have two questions. The first question regards the latitude we have on this committee, in that it seems that you're quite interested in our conclusions about how we might best use descriptors like race, ethnicity, genetic ancestry, or others. My question is, do we also have the latitude to recommend against the use of any particular descriptor or classification system? So for example, if we came to the conclusion that ethnicity was not a particularly useful classification system in genomics research, do we have the latitude to make that, to advance that claim?
I'm going to look to LSA or I'll look to anybody from NIH who wants to answer it, but I wonder if LSA or Vince want to jump in first.
Well, that's kind of quite a question to start off with, right? You're going to dismiss me in this context, as I don't identify as particularly with a racial group, as a mixed population, at least the population I come from as Latinos. I wouldn't know what the rules are, so I wouldn't know what Nassim or Eric wants. I think I heard some things that I'm on this from a general perspective, and in MHD, I'm not a geneticist. Everybody knows that, and this, as I said, is taking a narrow perspective. So I think if you broaden it, is one thing. I, for example, don't really understand how we can discuss all these things without acknowledging or discussing racism. I don't get it. And similarly, I think we have to also be realistic about what's going on in the clinical world, because to say we're not going to say anything about the clinical world is one thing. To say we're not going to say anything recommendation-wise, but we can't ignore it. Because it's a hot topic right now with a lot of groups. And the third thing I'd mention is, how can you separate this from social class? You're a sociologist, so I think that would resonate with you. These things are not living in isolation, and maybe it has less to do directly with ancestral analysis because of your social class doesn't influence necessarily your geographic origin or what kind of alleles you inherited. But I do think that in the big picture, in thinking about how we use these data to advance knowledge and to address issues, then I think that's a really important part of it. So
I'll follow up. The focus of this report is on study design. And so if this committee says this, let's say geographic location should not be used in study design, that's within the clear guidance that I understand that you have. So it's about scientific design and thinking about how can we improve the scientific design of genetic and genomic studies with regards to how populations are described. I think that the issues, and I assume the conversations within this committee would clearly bring different perspectives, whether that's a good thing, a bad thing, is it realistic, all the kinds of questions about that. But what we're looking for from this committee is to help the community with the quality of study design with regards to genetics and genomics research.
Thank you. I think we're very much on the same page on that. And then that will bring me to my second question, which has to do with the item in our statement of tasks, which asks us to propose culturally responsive methods. So I wanted to ask you for more clarification. What do you mean by culturally responsive? And since you also mentioned that you're looking for culturally responsive methods, not just in the US context, but globally, and we know that globally in different cultural contexts, there are very different stances on the inclusion of data on ethnicity, race, and so forth. I think we could really benefit from some more guidance from you about what you're looking for, what you mean by culturally responsive. Thank you.
I'll start and give it over to my colleagues. So I just want to first acknowledge all 14 institute centers and offices, and that many of these individuals are listening and watching this webinar today, but we really could only have a small panel to answer questions. But I think there was a clear recognition that came from some of my colleagues that we must be sensitive, particularly when you think about more in a global context, as well as within a US context, of how people see how they're described or how they participate in study. So a recognition that we live in a social context of our world. And so the culturally appropriate is really this question of recognition that there are different views and perspectives of how people see their identity. And that is important at times with regards to research. And I don't know if any of my colleagues would like to add to that.
Thank you very much.
All right, great. Thank you, Anne, and thank you, panelists, for responding to those questions. I think the issues regarding the global applicability of this are things where I think we'll be talking about more as the community does, the committee does its work. Now we have some questions regarding the exclusions. So you have given us 4 exclusions there, and there are some questions surrounding those. And I'm going to have Dorothy is going to ask the first question regarding those exclusions.
Great, thank you so much. And thanks for inviting us to ask our questions and for the helpful panel discussion that we had. So Eliseo already raised this question, and maybe others would like to weigh in, but I also was wondering about the beyond the scope exclusion of racism in science and genomics. Of course, a big part of our work will require understanding the meaning of race. And I think it's very hard to understand that without also taking into account racism and the relationship between racial categorization and racism historically and currently. And so I and the committee would like more clarity on the distinction between examining racism in science and genomics and what we can do to understand the meaning of racism, of race, in order to fulfill the task that we have before us.
So let me try to start and see if others want to extend what I said. You know, I don't think, if I heard it correctly, I don't think the exclusion meant you can't talk about, you can't understand, you can't have that be part of the formulation when you go to make recommendations about study design.
Okay.
I think what we were trying to do, out of concern that if you tried to bite off too much, it'd probably be nearly impossible to, especially on the kind of timeline and you're all very busy people. So, you know, LSA used the word awareness. I mean, I completely agree with that. So it seems to me, I think these exclusionary areas are areas we don't believe it's practical for you to make significant recommendations to fix. The recommendation is supposed to be about improving study design. But in order to make thoughtful recommendations, there probably needs to be a discussion and a very sophisticated awareness of some of these outside the scope for recommendation areas, if you know what I mean. That's my interpretation of it. I don't think we, for a minute, think that you have to put blinders on to these other critically important areas. They're completely influencing it. But we just didn't want to, we don't expect you to fix racism in the world over the next year. You know, but so that's my take. But let's make sure others from NIH agree with that take.
This is Stephen. I completely agree. I think one of the challenges is, as you come up with recommendations, they're going to have very far-reaching, implications. And I think thinking that through and accounting for that when you lay down your recommendations are going to be really crucial. We're not going to solve them with those per se, but we're talking about the language that we're going to use and the perceptions and the ways that we're going to conduct science going forward, and that could be really transformative and hopefully in good ways. But we have to worry about people taking the information and doing what they want with it. But I think what we're really hoping is that the research foundational aspect of this will really be solidified, and that we will have a way of understanding and a way of communicating that then allows us to both look backwards and, more importantly, I think, plan the studies of the future.
And this is Sherry Shelley. I just wanted to say sorry if my presentation came off as exclusions. It's actually just outside the scopes that you guys really had a scope to focus on, but certainly these themes and topics will be coming up in discussions.
All right.
But I want to.
Oh, go ahead, Eliseo.
Yeah, I just want to add the context. I mean, I thank you, Eric, for that clarification, and Sherry and Stephen. I get it, what the scope and what the plan is here, but it clearly, whatever the recommendations are, I don't know what we have all imagined might be your recommendations at the end. But, I think for me, it's just basic, simple things like, gee, I wish people would measure things the same way. And that we'd all agreed that it meant something, because up until now, we haven't had it. It's been, completely discipline directed, or completely do whatever you want. But racism has shaped this field from the beginning, and it's not a US thing only. So I heard the global component being mentioned. I thought that was pretty, widening the scope. We weren't trying to narrow the scope, but we just widen it to the whole world. And racism is very much part of, Latin America, for example, where I'm originally from. And it has been, very much core to many parts of the world. So this idea that it's built on, and it shaped these categories. And so what does all that mean? And I think that the committee does need to grapple with that. Maybe, we're not going to fix racism. No, that's far from our abilities here in our lifetime anyway. But certainly I think it needs to be factored in a substantive way.
Thank you. That's very helpful. Thanks.
Yeah. And thank you all for those responses. And whereas this committee's work may not fix racism, it's going to take a lot to do that, but I believe it can inform what we do in our work to dismantle racism. I think as Eliseo says, genetics and race have been tied for a long time. And I think what we do with this work can inform how we move forward, not just in genetics, but more broadly with regard to race and racism. So thank you for those. And Sherry, thanks for clarifying our language there as opposed to calling them exclusions, right? It's what is in scope and what's out of scope. So another question we have from Rick regarding some of the scope questions and the tasks that we have. Rick?
Yes, thanks, Charmaine. And I want to thank the sponsors and the National Academies for bringing this all together today, because this is a very, very important topic, and it's very timely also. So my question is a very specific one, and I think we talked a little bit about it with the previous questions, but the committee would like to further clarification in terms of what appears to be a conflict between tasks 2 and 3 and the beyond the scope exclusions of 1 and 3. And so specifically, this issue of not dabbling much in this issue of healthcare, health-related research, but at the same time, you're asking us to provide some guidance or recommendations around genetic risk scores and the disparities related research. So just some further clarity on that would be helpful.
So I'll take the first pass. It was my understanding that what was out of scope was healthcare, not clinical research. Is that my NIH colleagues agree with you? So in which case, number 3 would be fine, because you'd be looking at, assess the appropriate use of race, ethnicity, genetic cancer risk population, and the determination of genetic risk scores and health risk. And maybe we should have added a phrase within, clinical research studies, or clinical research studies examining genetic and genomic risk scores and health risk. Vance and Sherry?
Yeah, to expand on that, this, as you know, this is a really important issue. And actually, I think there's other efforts going on at the National Academies dealing with some of the broad issues about race in medicine. That the intent here was to keep this focused on issues around population descriptors in research. So that includes polygenetic risk research, right? And how populations may get described with regards to perceptions of polygenetic risk. But what was intended was not to get involved in issues of thinking about algorithms and clinical guidelines, the appropriateness of use of race or ethnicity as factors related to clinical decision-making, which are important issues. are issues that clearly over the last couple of years has gotten much more of a focus. But this study is focused on issues with regards to use of population descriptors in research itself, including polygenetic risk. So that's the distinction that is seeking to be made.
And one prototype, I'm not claiming it's the only prototype. I mean, one prototype is my institute recently launched a major new consortium that's looking at the use of polygenic risk scores in different groups to address exactly that. And they will be struggling and tackling these issues within the context of a major clinical research study. They'll have to be describing this, their population and so forth. But that's different than clinical practice guidelines for healthcare implementation.
But let me add to that. I think that the emphasis, we've used the word sort of foundational or etiologic or basic genomics and genetics is really crucial here. And how that has a gray zone towards clinical care is often very difficult. And you'll have to think about those questions, but we're not asking for the recommendation of how to go to the FDA and say, this test should or shouldn't be approved for these reasons for a given group of individuals at this point. Now, the FDA is going to be watching what you're going to be doing because it's going to be of interest to know where that science is coming from. But this is not about recommending the actual final products or drugs or whatever based on that. But the language and the way in which we frame the science is really crucial and will have tremendous implications downstream.
Thank you.
Thank you. Thank you all for those responses. So I have a question regarding the number 3 point under what's out of scope. And that there we talk about one of the item there is examining the use of race and ethnicity in biomedical research generally. That's out of scope. And we were just wanting some clarity on the difference between genomic research and biomedical research that includes genomics. So how were we thinking about genomics research? I mean, clearly genomics research and biomedical research are connected and in many ways intertwined. How are we making that distinction between genomics and biomedical research that involves genomics?
Vince, do you want me to take the first pass at this? Or?
Yeah, you can take the first, and then I'll follow you.
Here's my thinking, and I admit there's so many smarter people on this committee than me about some of these issues. So, I think some of what you're hearing is we just didn't want to burden this committee with the ocean. I mean, it was just going to be overwhelming. I mean, these are huge problems. And what I think that limitation very much goes back to this workshop we did, I think it was 2016, if I remember, I know it was on my time,
yes,
that we did with LSA's Institute. And we just said, let's just take this slice of the biomedical research problem because it's just a slice. And, maybe it's my incredibly biased view, and I admit I'm incredibly biased now being into genomics for 35 years of my life. The last it's just incomprehensible to me that in studies where you have significant amounts of genomic data, which gives us incredible amounts of insight, that we end up going back to population descriptors that get us in trouble, that seem incredibly 90s, or even worse. I mean, it just seems that we overwhelmingly burdened ourselves in very unsophisticated ways at a time, and I'm biased, that we're getting genomic data, which is giving us the most cutting edge, sophisticated insights about these individuals. It's just that lack of harmony between those two that led to that workshop and has led to my thinking there must be a better approach. And the better approach, if we could figure it out first, what I think is almost a best case scenario with a lot of genomic information, maybe we could have lessons learned to generalize it. So I think that's what drew me into getting very excited about this, but I'm sure there's other views, but it just seems that let's first solve this, or at least first think about it in a limited way about those studies where you actually have genomic data and deliberate whether that will or will not be helpful for this. I tend to be thinking it could be incredibly helpful, but that's what we want your input about. Vince?
So just to add on that, when you think about genomics, it's integrated now across all biomedical research, right? And the questions about how population descriptors there will ultimately impact other areas of biomedical research. But there was a goal here to keep this focused so that we could clearly develop some best practices related to the field of genomics. I think that your recommendations are going to have impact on studies that people are never doing any kind of genomic analysis whatsoever. And I think that's important, but that's not the charge. The charge is for you to stay focused on genetics and genomics and how descriptors are used, but clearly recognizing that it's all integrated. But we were really trying to give the committee a focused area so that the report can have a great impact within that disciplinary area that crosses many different disciplines, but is clearly focused on people who are doing some type of genetic or genomic analyses.
And to be able to do this in a year. I mean, if we think about if it gets too expansive, doing this in a year, more, it's going to take much more than a year.
So can I ask a question of clarification? We have quite a few others, but I guess the question is that so many genomic studies are motivated by biomedical questions themselves. So I think it's going to be very difficult to sort of not disentangle them, but to do one study without really figuring out the effects in biomedical research generally.
Yes. Great point, Aravinda, because I'm still trying to figure it out myself. But
I guess the one comment I want to make, kind of following up on that, and really love to hear from the committee, is, I mean, that is a broad range of type of research. If you talk about biomedical research and think about it in that broad context, and wanting to actually help to move the fields of science forward. And so from that perspective, I'm personally concerned that we will not have the impact with regards to your guidance. And so by focusing in on genomics, I think that you have the potential to have great impact, both on the broad fields and studies of genetics and genomics, but ultimately other areas. But by not taking on the world, I think the potential of this report is greater. That's my personal perspective.
Or use it as a first step. And if it's wildly successful, then the next year, I could imagine another phase of this. But we just were so concerned you would get burdened down with so many nuances and little nooks and crannies across biomedicine. And I will also point out, one might have argued, or one might have expected that with such a limited scope, it would just be the Genome Institute, my institute, willing to support this. But I think across NIH, people recognize this was maybe the best approach was first try to have this looked at through this somewhat narrower lens of genomics, and certainly lots of other non-genomics oriented institutes, although everybody's doing genomics, they saw the value of that.
Yeah, I think there's a point, Eric, in terms of your side comment there that everybody's doing genomics. So thinking of biomedical research that includes genomics.
Yeah.
We're not talking about all biomedical research, but even if we're talking about biomedical research that includes genomics versus, and sometimes it's the investigator, whether that's investigator is thought of as a genomics researcher or a genomicist or geneticist versus other researchers doing biomedical research that include genetics. Is that part of the charge? Yes.
Yeah. I mean, once you have genomics data, genomic information in the mix, it's in scope.
Okay. All right.
Okay. That clarifies it. Thank you.
Yeah. I mean, maybe another way to, so maybe if that helps, Charmaine, it doesn't just have to be NHGRI funded research.
That's right, Eric.
That helps. We apply that.
Yeah, we weren't talking about all biomedical research. We're talking about biomedical research that includes genomics, but that ordinarily may not be considered genomics, if you're thinking about genomics in a narrow sense. So.
And I think that's important. So even think about social genomics, social scientists who are doing genetic and genomics analysis, that's within scope. Included. That's within scope.
All right. Glad we fixed that problem. Thank you. All right. Mike has a question for you, committee, for you panelists. Sorry.
Hello, everyone. Given that the task list charges the committee with delivering guidelines, best practices, and implementation strategies about the use of population descriptors in genomics research, the committee wondered why making policy recommendations directed at NIH and other government agencies was beyond the scope.
So I'll start on this one and ask my colleagues to join in. We wanted to make sure that this was a report to the scientific community about best practices. And so that the intent of this committee is not to think just about NIH. Now, by giving those guidance and best practices, that will inform NIH, but this was intended to help the scientists, the researchers, the academic institutions, the research institutes, the industry that's involved in these work to help with best practices versus a report to give specific requirements of what is needed to move best practices to through the NIH.
Okay, thanks, Vince. Anybody else wants to weigh in on that question?
This Eliseo, I would just, just in general comments, reacting to some of the discussion, I concur completely with, Eric's sort of guidance of, we don't want you to sort of boil the ocean kind of thing, because this is never ending conversation otherwise. Plus the assembled committee, I think, is not necessarily doing clinical work or involved in as much clinical research as other groups might have to have added that level of perspective. But the report will be closely watched by all those groups. So I think as you deliberate and review documents, some thoughtful reflection and consideration on these issues that are quote unquote a little bit out of scope would be welcomed. I start from the premise at NIMHC, and I say this almost every time I speak to any audience, that this self-identified social construct of race ethnicity that we call influences health in so many ways that we don't understand, that we have to measure it and we have to consider it. And I think that's true for genomics research, for biomedical research, for social science research, and for clinical research and for clinical care. Until maybe someday it won't matter. I don't know, but right now I think that's universally true, and it has this major component of social class or socioeconomic status that is similarly a pillar in this involved. It has no genetics connected to it necessarily, unless it's interactive. And what we already discussed around the issue of the history and racism and discrimination sort of leading to these categorizations in order to create inequality, and we can't ignore that in going through this report. I mean, I think the charge is clear. It's narrower. It's going to help the field. But you're all smart people. That's why you're here. You're going to have to think about, weigh all these options, all these considerations as you proceed with your year-long effort. So thank you.
Thank you, Eliseo. And it's when and how and why we use these variables in many cases. Yes. Molly has a question for you.
Right, so mine was quite a specific question. I was just reading the points, and I was struck by the fact that in point 6, you state that we're charged with assessing when it's appropriate to use race and ethnicity as population descriptors, and you don't include genetic ancestry in that question. And I was just trying to understand whether that was deliberate or whether, in fact, we're being asked to consider when it's also appropriate to use genetic ancestry.
I'll start on that one. That was not deliberate. So it really is broad population descriptors, how you deem appropriate. And I know the issue of genetic ancestry is clearly an important topic area. But as I said earlier, you could be talking about geographic, the appropriateness of using that. So whatever population descriptors that you deem appropriate to be part of your consultation.
Thank you. Thanks for the clarification.
Thanks. All right. Anybody else wanted to weigh in on that? All right. Let's move. Arvinda, you still wanted to ask your question?
I think Sandra should go first.
All right. Okay. So let's do.
I'll ask after that.
All right. Okay. Sandra?
Yeah. Well, thank you for answering our questions about the charge. As we think about our work, particularly considerations around implementation and barriers to new approaches, and I'm thinking here about how they impact existing data sets, I'm wondering to what extent is our charge to reconcile past practice in the use of racial and ethnic categories, and I'm thinking legacy data sets, when we think about future practices. And this seems particularly relevant for number 4, 7, and 11, and the desire for harmonization across time and place with respect to large data sets.
I'd happily jump in on that one because I think that, thank you, it's a really important point that you bring out that I think we've stated in several different ways, but it's really going to be important to understand how your recommendations could be applied to legacy sets, because we know that there is a tremendous amount of information, and this is where it goes to the genomics that are available and the like, that how you would use different tools or different ways of potentially reinterpreting, reanalyzing, or rethinking that. And that's going to be really important to understand how to do that, because what we don't want to do is forget genomics for the last 100 years and say, all right, now we have something going forward only. And that's probably going to be one of your biggest challenges in giving some recommendations about that.
So squarely within scope is what I'm hearing.
Yes, I mean, in many ways, it's really what so many, I think, in the research community are going to want to know what to do with my existing data. Right. How should I look at it differently, or how, this may have real implications. We haven't mentioned, scientific editors, but you could imagine if the recommendations go a certain way, you could see the editors are going to be saying, oh boy, we have to go back and, restate or ask people to look at or, republish their data in a way that's more appropriate and more useful going forward. I mean, that's the medical and scientific publication is clearly going to, I think, show up in your discussion, and how and what ways you would have an impact on that are going to be really very important.
Thank you for that, Stephen. Anybody else wanted to weigh in on that question from Sandra?
I would just say that this was an area that many of the institute centers and offices saw as an extremely important area. And we recognize this is going to be a challenging area for the committee, but your guidance will be extremely important to the field.
Thank you, Vance.
Can I add one other point? I think
sure, Stephen.
In going to the legacy data, it's really going to raise a really important question about past data sharing and data availability, and looking at that and thinking going forward. There's a new policy the NIH is putting out in 2023, and that will continue to be modified. But I think your discussion may raise some really interesting questions and thoughts about what to do about data sharing and being able to address and have others understand and validate or confirm what is a recommended approach per se. So.
Right. Great. Thank you, Stephen. Any further comments from the panelists on that?
Yeah, I wanted to raise a couple of things just to make sure. I think all of you have given us a somewhat better sense of what you intend by scope and limit, but surely it's difficult to, I mean, we have to bring up other issues that will inform the debate. In fact, it would be, I think, not possible to even answer your primary questions without addressing all of those issues. And some of them are very hard issues of racism and so on. But hopefully and actually more than hopefully, I'm quite sure that the panel, that the committee will think of these issues, and I can tell you we'll do the best job we can. A few others, I think, when you talk of committees, it leads to my question, is I presume this will have some impacts on what the nature of consent forms look like. I assume that this will have something to do with issues of what journals and already I know that some journals have reached out because they're interested in what's going to happen. But the one thing you didn't mention, and which is that you talk about population descriptors, but of course it also means individual identification. Populations are made of individuals, and you can't just label whole groups of individuals without at least individuals having some label that they agree with or not agree with. And we will have to, of course, debate and think of it. But ultimately, we do this as a very broad group. Who's the report for? I mean, is NHGRI going to, I know you didn't ask us to engage in policy, but we will try to stay away as much as we can, but I know there'll be recommendations of what NHGRI and the other institutes should do. But who is this report ultimately for? I recognize it's going to inform a much larger audience. Is this gonna, you don't know, or is this gonna come to grantees to the 14 institutions saying, this is what you're going to be bound to? Or because, it's possible for some of these, we may not have a very adequate answer. We may not have a very practical answer. And I don't mean to say that we land up there, but I'm just trying to understand, is this what this report eventually, who are the primary consumers of the report?
So let me make the first comment, and then I think Vince probably should carry on. First, because I want to immediately dismiss there's anything unique about NHGRI in this. We are equal partners and equal everything with our other 13 components of NIH. That said, it's conceivable it'll be used in different ways by different institutes and programs, and potentially by different groups outside of NIH. I think Vince said, rather decisively, the purpose of this is for the audience. The main audience for this is the scientific community for the purpose of the design of research studies. That's not to say that it won't then be used as information for building out other things, whether it be policy, whether it be grants policy, whether it be other policy, whether it be journal policy, so on and so forth. So I don't think we know, nor do I think we're in any position to limit that. But I would say first and foremost, it is for the generic biomedical researcher, of which we're the most familiar with the NIH-funded ecosystem, but we make it widely available to anyone who wants to use it. But to my knowledge, no part of NIH is sitting there and saying, once we get this report, it will be put into action in A, B, or C. I don't think we know, because we don't know exactly what the report's going to say. Vince, what would you like to add?
I would just agree with that statement and say this is for the scientific community, for researchers who are conducting genetics and genomics research as part of their whole research program. And that this would be a consensus study that they could go to help them as they are designing their new studies and their work. That is the central target for this report.
Okay. Anybody else wanted to weigh in on that from any of the other institutes? Yeah.
This I'll say, I'll just say that we can't really require our grantees to do anything, right? Sometimes
you could ask them to share their data. That would be a good place to begin.
Right. There has been evolution on some of these elements, data sharing being one, and common data elements, which was mentioned earlier. another one. And it's not easy. I mean, people are very, wedded to their way of doing things. And in the field of population science and social behavioral and clinical research, it's probably even harder than it is with the basic researchers in order to do this. However, we can sort of say, well, this was a report, this is the way it's endorsed, and these are the way we want you to do it, and encourage everyone to do so through, links in our funding opportunity announcements. And at least I'm maybe delusional, but I'd like to think that gradually we'll be changing the paradigm by doing that. And we've already embarked on that in the area of using common measures. But I think that would be the way I would see it. So.
All right, great. Thank you for that and for answering our questions. If any of the committee members have any other questions, feel free to raise your hands because we have some more time. We can grill the panel a little bit more if you have additional questions. I know Katrina has a question. Go ahead, Katrina.
Charmaine, thanks so much. And it's great to see everybody. I will say, I think the whole story in academic medicine LSA is that you can't make anybody do anything they don't want to. So I guess my question was a little bit, that this report is coming at a time where I could imagine that there is a body of science, of evidence that would be reviewed, and you could imagine specific recommendations being made. At the same time, I think we all recognize that all of this is just the most unbelievably dynamic area. Probably that's true for all of science, particularly this area. And so over time, I've seen these reports balance between trying to kind of capture the current state of the science and what one might do today versus recommendations about what might happen in terms of a more dynamic, ongoing way of creating systems that would keep things more up to date or more reflective of changes in the future. And I guess I was curious, as you all think of what would be most helpful, what the balance, obviously it's often hard to figure out where those latter committees or whatever that would be would sit. But I'm curious about the balance as you thought about this report.
Not surprisingly, that is an incredibly good, insightful, but difficult question to answer, Katrina. I will say that, when we suggested names for the committee and thought about who the National Academy might put on this, that included people who we thought were actually even better positioned, even by far than some of us, certainly me, to really be able to answer that. I mean, in part, it's how well can you anticipate where things are going to be 5 years, 10 years from now in some of these highly dynamic research areas of understanding population diversity, population histories. I certainly could just represent genomics, knowing that it's constantly moving so fast that it's always hard to make any decision because any decision or anything you say is going to be outdated in a matter of months. So I think this is something that I don't mean to kick it back to the committee, but I think that's why we were thrilled to see the composition of this final committee the National Academy selected, because it included some of the most and brightest minds in this area to sort of help determine, how far to reach forward, what might be possible in 5 years, what recommendations would be durable in the face of current trajectories of some of these research areas that maybe would make it so they don't get outdated in 2 and 3 years. So I don't think it's an either or nor. And I think, like a lot of times in these things, we have to try to thread the needle. We would love to see your recommendations have a shelf life of more than just a year or two, but we also don't want them to be so hypothetical and contingent on advances that we don't know for sure are going to be there. That would be my best answer to a very good question. I don't know if others want to weigh in from the NIH side.
Thank you, Eric. Yeah, anyone else?
Yeah, no, this is Stephen. I think that's a terrific question. And in looking at this subject matter, I think that should be a big part of your charge. I mean, you look at other things that, for instance, the program that I run, we come a lot of times to the National Academy of Sciences over epidemiologic or risk factors of exposure to radiation or chemicals in the environment that we know we are changing in how we measure and understand those. And the reports are not about what is the cross-sectional understanding on that day, but rather how the science is moving and what can be done in the future to be sure that those questions continue to be studied and refined as more data comes in. And so I think that I wouldn't look at this as a cross-sectional analysis. This is more prospective. I mean, what is it that we can understand from you all about both the principles and the behind the recommendation, as well as the recommendations that would be useful for funders and for academics and people who are doing that, the basic genomic research in 2022 and 2025. We know it's going to continue on. So I hope that that's part of your, part of your mindset as you lay out the recommendations.
I just want to add, I agree that this is a central question you just asked, Katrina, and I hope that as the committee does its work, it's thinking about that issue. One of my colleagues from another institute just said, we want you to help us how we can think about improving science. And so thinking about both today and in the future is extremely important. My last comment on this topic is that as I look back at some of the seminal reports from the National Academies, they were dealing with a problem of that day, but they were also looking forward and continue to be of value, years later. And so that's my hope with regards to the work that you do.
Thanks, Vince. Anybody, Eliseo, others want to weigh in on that question?
I think, Katrina, it's a framework, right? And it's a method. And so as long as you take the approach that you're not setting dogma and saying, okay, here is this way or no other way, I think this committee recommendations can have a long shelf life and guide us for years to come. I mean, after all, we've been living with some of these categorizations for decades and not find two people that love them.
Yes, thank you, Eliseo. Sherry, did you want to add anything to that?
No, I think my colleagues have said it quite nicely.
All right. So I'll ask my colleagues on the committee, anybody has any other questions? And then I'm going to give our esteemed panelists the last words before we end this open session. So anyone else from the committee has any other questions for the panelists?
No, but as a co-chair, I think we're all aware that we may seek further clarification down the road. Yeah,
that's right. Absolutely. It's not speak now or forever hold your peace.
And Aravinda, I'm sure you heard me say that, and it was very genuine. And I speak not only for myself and Vince from NHGRI, and I'm sure the other panelists you're seeing, but there really are at least 10 other, probably more than 10. There's representatives from the other 10 components of NIH listening in on this, and from all nooks and crannies across NIH. And there is just a considerable amount of goodwill, enthusiasm, excitement, and willingness to help you from across NIH. So I hope this committee, as they dig in deep, come up with more questions, come up with any things you want feedback, we will get you that feedback from NIH from any part that you need it.
All right, thank you, Eric. And on that note, my question to each of you, and I'd like to hear from each of you. As we think, Eric, when you gave your presentation, you talked about this potentially being a landmark report. Potentially, we hope it will be. But want to hear from each of you beyond the statement of task that you've given us, and we've discussed that in quite a bit of detail. What else would you want to share with us about your vision, your dreams, your hopes for what we will do? But even guidance as we do our work, additional guidance that may not have made its way into the statement of task. And I'll let Eric start if
Sure. Well, let me first address the second thing. I think a lot of this discussion about scope, what's in, what's out, what's genomics research, what's not genomics research. I think the reason we are all struggling with this is that NIH really wanted, they really wanted a practical a study that could practically be completed by the very busy group of you in a year, and that would had a practical or realistic ability of being impactful because it was practical, as opposed to trying to be too big or too small. So I think if you're struggling with that, as did we internally before we ever got all this out of the gates, it was just trying to thread the needle between wanting to fix all these problems, but at the same time recognize you can't fix all of them. And so just let's start with a good solid step of having this be more limited and have some boundaries around it. So I think that's what we are all struggling with, but I hope you realize we're doing this out of goodwill, that we're making people struggle with this is because we like you want this to be successful and impactful. You sort of said at a personal level, any other things to add to this? Again, I come to this, I'm looking around the room. Yeah, I mean, more than anybody else, my blood runs, 99.99% genomics. I think the other people on the panel have more diverse scientific backgrounds, bring other lenses to this. I have seen how genomic advances over the last 3 decades have laid clear the problems in this area, and I mean, I almost, just absurdness of what we do and how impractical it is in so many ways. And I guess my hope is, and it may be a naive hope, is that genomics can actually be part of the solution here. Is that somehow maybe genomics could be used in a leapfrogging kind of way where we could sort of not be encumbered with some of the other very complicated histories associated with some of these words and some of these frameworks. And maybe genomics provides sort of a leapfrogging type of capability. I don't necessarily know exactly what that looks like. I think some of you have expertise in this far more than I do, but that's what I might like to dream about, that we could see a part of the solution being a genomics-based solution.
Thank you, Eric. Thank you so much for that. Anyone else wants to jump in?
Yeah, I was just going to say, from the perspective of the All of Us research program, certainly genomics is part of what we're doing, but we're bringing in all different kind of data. And so I think this is really setting the stage for what's to come in the future. But the in scope and out of scope stuff really is to put some guardrails on what this study in particular does, and we can always grow from there. So just wanted to make sure that you guys know that this isn't the end, or it may not be the end, but it is the start of something big.
All right. Thank you, Sherry.
I'd like to add something. I'd like to have a different kind of charge to the committee, thinking about the importance of this report that will be quoted or used for decades, because I think there is that potential. I mean, we've had workshops and we've talked about it, and you've seen things written in the lay press and particularly in the scientific press. But this group could come up with, and we hope, something really concrete and useful that would be referred for years to come. That happens with Academy reports. It happens with Surgeon General's reports that really crystallize and help the field to move forward. And so it's really our hope that you all will be able to come up with something. And a year is a short period of time, but and by asking to do that in a year, that's telling you in an indirect way of how important it is. We're not interested in waiting 2 or 3 years for the solution, because it is that important. So don't mean to put any more pressure on you than you're already feeling, but recognize this is really a potential transformative moment, and we hope that you do what you think makes sense in considering everything, not necessarily saying that you can't do this or you can't do that. This really is a remarkable moment, and we hope you sees it well.
Thank you so much for that, Stephen. Appreciate those comments. Vince Eliseo wants to jump in there.
Well, no pressure at all from Stephen there, right? I don't have any major wisdom to part you with. I think fundamentally, I see these critical areas, constructs, as tools for discovery. And I think if you set up, a nomenclature or a categorization that allows that to happen, so we're not getting confusing perspectives on, what does it mean to be of this particular group, have this particular profile and this particular outcome, I think that will go a long ways to being really transformative. And not being, we're not making definitive social statements here, although there will be many out there who will try to make that out of what we say or what you say. But I think keeping that perspective allows us then to manage the sort of the waves that come with it. So again, I appreciate all the work that you're going to be doing.
Thank you, Eliseo. And Vince, you get the last word from our
so
sponsors.
I want to just start by thanking each of you for making this commitment to be part of this committee. This will be a lot of work, and I know it's going to take a lot of time, but it can really make a difference with regards to biomedical research and particularly genomics research. And so I just want to thank you for the commitment that you're making to this. The one comment I would want to leave with you is I hope that you truly engage diverse parts of the community in the conversations. I know you'll be having 3 meetings. I hope you have general public. I hope you have journal editors. I hope you really reach out to engage the communities. And my hope is that because of this committee and the diverse disciplinary background on this committee, that scientists will truly seek to understand your recommendations and use them as they conduct their research. And so that is my hope, but I want to just thank you for your work, and I look forward to seeing the recommendations.
Wow. Thank you, Vince. Thank you, all of you. Thanks, our sponsors, for taking the time to talk with us and for sponsoring this critically important study. I'd also like to thank the audience for your interest and for joining us today. We look forward to having you at our future open sessions of the committee. And a special thanks to the National Academies staff. Just been, the work is just beginning in terms of our first meeting, but they've been working on this for quite a while and working really, really hard. And we appreciate everything that the staff has done, Sarah and Andy and Leanne and the rest of the team that we've been engaging with over the last several months. So thank you all. If no one has anything else, our open session is about to close. So any final words from anyone, committee members or anyone? All right, well, thank you all committee members. Remember, we reconvene tomorrow. So we'll see you again tomorrow. Looking forward, and thank you everyone for being part of this process. And it's exciting, and we look forward to seeing what is going to come of it. Thanks.
Take care. Be well.
Bye,
Thank you, everybody.
See you tomorrow.